human decide who will receive what traits and who will receive what traits. DNA is a long, thin strand of molecules that carry genes and the codes that make people who they are.
First, let's start by explaining what DNA is actually made up of, so you are able to understand how it is structured. It is made up of 4 different nitrogenous bases called adenine, thymine, guanine, and
cytosine.
It is also made up of deoxyribose and phosphate molecule. I know you're wondering how anybody could ever figure this out, and it was not us who did. The people who discovered this were named Watson and Crick.
The bases resemble the alphabet and can be arranged in almost an infinite number of orders to create combinations. Each mixture of letters is a trait on the strand of DNA, which permits for complete diversity among humans. This is how we give the citizens of the city their unique characteristics. Emma and I have special wands that look like strands of DNA that are shaped like a double helix or a windy ladder-like object. We decide on the genetic code of the organism, and then we use our wands to give the codes to the babies when they are born.
Hey Jillian! I think I hear the alarm going off! You know what that means! Let's go to the baby!
Ah! My baby is coming! My baby is coming! Ah! I just had my baby! Okay. She's so cute!
Emma, let's look at the parents' genotypes and give the baby some traits.
Let's do this! So, both the parents have blue eyes and blonde hair. So, that is the baby that is going to be, right? Easy enough.
Now, for the rest of the traits, it is quite complicated, so just watch and learn. You have to find the alleles that each parent has and find the combination that the parent is that the baby is going to inherit, and voila! You have you have a baby with some pretty traits.
Aw, my baby is so adorable! Thank you, DNA heroes!
Just doing our job.
Well, Michelle, that isn't the only part of the job. DNA also makes the proteins that we need to make traits, and it's called protein synthesis. There are a few basic steps in the synthesis of a protein. First of all, there's transcription of mRNA, which is the DNA copy from a DNA gene in the nucleus. Next, in the cytoplasm, the protein synthesis is started by mRNA, and it travels through the ribosome 3 bases at a time. Finally, the tRNA, which is the transfer RNA, matches up with the open DNA bases, and the tRNA releases the amino acid at the top, which joins the chain of amino acids being produced.
And there you have it. That is how proteins for the traits are made.
Now, Michelle, you might be thinking, since mRNA is a copy of DNA, they must be identical, right? That's where you're wrong.
Really?
DNA is always found in the nucleus, whereas RNA can be found in the nucleus or the cytoplasm.
Wow!
Also, DNA sugar is deoxyribose and its bases are ATCG, but RNA sugars is ribose and its bases are AUCG. So before you go and call them identical, remember that information.
I will.
Michelle, this baby looks very healthy, and fortunately, the DNA that we gave her majorly contributes to her future growth, as well as helping her cells to function. As well as coding for proteins, DNA provides the genetic blueprint of an organism's life. The DNA allows the cells of an organism, in this case, your baby, to come together and cooperatively create a well-functioning living thing.
I love your magical wands. What are they? Are they chromosomes? I've heard of that word.
No, silly. Our powerful wands represent the gene strands that are given to organisms.
Wow!
Chromosomes, however, are the structures that hold the strands of DNA. That DNA is bundled into thread-like features, which are chromosomes. And each and every chromosome contains a structure called the centromere, which separates it into 2 different segments. The chromosomes, as well as genes, which contain the DNA, are located in the nucleus of the cell.
There is one more thing that Julian and I want to inform you about that's very important.
Cass, thanks.
It is in regards to occurrences that can take place that are not necessarily supposed to. Occurrences are called genetic mutations, and two examples of types are insertions and deletions. In an insertion, DNA is added to a gene which changes the number of bases.
Caught it.
This could cause the protein produced to not work in a proper way.
Deletion, on the other hand, is when a piece of DNA is taken out. Deletions range in size, and the larger the deletion is, the higher the chance of an entire gene or neighboring genes will be removed. The deleted DNA can also change the function of the produced proteins or proteins. Some examples of genetic mutation are color blindness and cystic fibrosis.
Thank you so much for helping us, me and my baby. I am so glad that I learned all of this new information on DNA.
You are welcome. We're happy. Job well done. See you next time on Superhero Headquarters.
Baby, you a song, you make me wanna roll my windows down.